My name's Rebecca Bullers, and I am a graduate research assistant here at MASARC. And today I'm going to be discussing my master's thesis project, where we are studying acoustic conditioning in common carp to accelerate removal and reduce costs. So I'll be presenting some preliminary results from our lab experiments that took place this past spring and summer, as well as our 2023 field season, which should actually be wrapping up in just a couple of days. So, as many of you know, common carp are one of the most widely distributed invasive species in North America. They have destructive feeding habits that consist of sucking up substrate from the lake floor, and then they have a unique palatable organ that allows them to release non-edible particles back up into the water column. And this contributes to the reef suspension of sediment and nutrients impacting turbidity, phosphorus levels, and other water quality parameters, as well as causing a decline in biodiversity and abundance of aquatic vegetation, waterfowl, aquatic invertebrates. The list kind of goes on and on. So their damaging impacts have led to an increased need for a safe, selective, and effective management strategy. Studies at the University of Minnesota have shown that we can induce these large feeding aggregations of carp in the summer by using species selective bait, such as cracked corn. So carp are highly social fish, and they can learn the locations of these baited sites in just a matter of days through group learning. So this graph above shows the number of unique fish present at a site each hour over a 7-day period. And bait was introduced at the site on the night of the 30th, which is denoted by that small little star. Um, and you can see that carp started utilizing this site just a few hours later. So these aggregations are typically paired with different physical removal strategies, and being able to control the location of where these aggregations are proves to be extremely extremely valuable, allowing removal to take place where practical. We're also able to increase the frequency of removal events and accelerate the control of carp populations with this incentive of bait at our sites. So they're repopulating these sites more quickly after removal events. Currently, one of the biggest limitations with bait and removal strategies is that carp visit the baited sites at different times throughout the night, and the aggregations vary in length. And current strategies are limited to the amount of fish that are present at the exact time of removal. So we're hoping to tackle this problem using acoustic conditioning, which in this context refers to simple operant conditioning, where we are trying to condition carp to associate an acoustic cue with a food reward at a specific location at a specific moment in time. So we're trying to accomplish this by using an underwater speaker and an automatic feeder that allows us to regulate when the cue is being played and when the bait is being administered. And this may be able to help overcome some of these limitations of bait and removal strategies by synchronizing feeding aggregations, allowing more carp to be removed at one time, further accelerating the control of carp populations and hopefully lowering the costs associated with bait and labor. So this has been shown previously to the start of our study that carp can associate food with an acoustic cue. So two papers that were influential in the development of our experimental design were written by Sloan et al., and it was based out of University of Minnesota, Duluth. And they showed that common carp were able to become conditioned and associate sound and food in both a laboratory and an outdoor pool setting. And they also showed that carp were able to localize the source of sound within that controlled setting. There was also a fairly influential paper out of Israel produced by Zion et al., where common carp were acoustically conditioned to an automatic fishing machine in an Israeli reservoir. And they were originally testing the response of a different fish species, so carp being conditioned was kind of a bonus to their study. So it has been shown in both the lab and the field, so we were hoping to replicate this and hopefully use it to synchronize our feeding aggregations. So if any of you were present at last year's showcase, I presented on our 2022 field season, which took place in Silver Lake in Ramsey County. So we had two sites, each with an underwater speaker and an automatic feeder. I'm not going to dive too much into the setup because I will explain it more in detail for our current field season. However, both of these sites were exposed to a period of sound and bait, sound only, and bait only. And when comparing those three different treatments, we confirmed that carp were responding positively to bait. However, the acoustic cue did not attract or deter carp from our baited sites. So we thought that these results could be due to a relatively low carp density within Silver Lake and the availability of other food resources. We did see a decline in overall attendance at both sites throughout the summer. So this led us to selecting a higher density carp lake this summer, where there would be more incentive to respond to the bait in a timely fashion and hopefully a better chance of them becoming acoustically conditioned. We also decided to run some experiments in Mazark's containment lab to further explore carp's behavior in relation to the acoustic cue, and then use that information to help plan out our protocol for the 2023 field season. So the main goal of our lab study was once again to replicate carp's ability to associate an acoustic cue with our food reward at a specific location. So we did this by conditioning them to approach a feeding ring on the onset of an acoustic cue where they would then be rewarded. But we also wanted to explore different baiting techniques to determine if carp would respond better to intermittent but larger food rewards, which we'll refer to as partial reinforcement, or continuous smaller food rewards that will be referred to as continuous reinforcement. So it's been shown in other animals such as lab rats and pigeons that they display stronger conditioning behaviors and a higher resistance to behavior lost when they have a reward that's applied in a discontinued fashion. So this is kind of a known theory known as the partial reinforcement extinction effect, or PREE for short. So we're hoping if fish undergoing this partial reinforcement treatment perform better or just as well as fish undergoing continuous reinforcement, it could potentially have management implications down the line and allow us to cut down on costs associated with bait and labor in the field. So our experiments took place from April 2023 to August 2023. And our lab space consisted of 10 tanks, each with three small common carp. They're on average about 40 grams, maybe 4 inches long. We had a UW30 underwater speaker. I'm gonna try to use a laser here. Let me know if it works. So that blue disc on the bottom is our underwater speaker. And then we had this feeding ring made out of plastic tubing that laid flat on the water surface. We had an automatic feeder that held fish pellets in this little groove here. So it rotated and dropped the food into our feeding ring. And then we also just included a black plastic mesh cover and tarp to cover up the tops of the tanks just to make it a more secluded environment and help the fish habituate. So during the trials, we also had GoPro cameras that were recording every single trial. We would control the cameras, the speakers, and the feeders remotely from a different location within the lab to prevent any human influence on fish behavior. So we conducted 2 rounds of experiments. So we've repeated our experiments in these tanks for 2 times. So prior to the start of the experiment, we sound mapped all 10 tanks to ensure the cue would travel throughout the tank and it was louder than the background noise of the lab. For any of you that have been in MCL in the past, it gets quite noisy in there. So we just wanted to make sure that the fish would be able to hear it from every part of the tank. So that's where you see the average background sound pressure level was 117 decibels. The sound pressure levels with our acoustic cue was 142. So on average, there was a 25 decibel difference. So the fish would definitely be able to hear the sound during our trials. And the acoustic cue that we were using was a 400 hertz pure tone that was 30 seconds long and it pulsed 2 seconds on, 2 seconds off. So also sound pressure is just the strength of the acoustic wave. So typically, the louder the sound is, the higher the sound pressure is. So these little X's just mark where we are taking our sound pressure measurements. And then we also took one right above the speaker as well. So our experiment was broken down into 5 different periods. And each period had 3 daily trials. So the 3 daily trials varied in start time by about an hour or so to ensure that the fish wouldn't have any sort of temporal association with the feedings. But the acoustic cue was played for every single trial, but the amount of bait and when the bait was dropped varied for each period. So we'll just kind of run through them one by one. Sorry that it's a little busy of a slide, so let me know if you have any questions. Um, but during the training period, which lasted 10 days, the food was dropped on the onset of the acoustic cue within the feeding ring. So this was just a period of time to help the fish associate the food with the cue and with the feeding ring, just kind of further helping them habituate within the tank. And then the next phase was the conditioning phase, and this lasted for another 10 days, and the food was dropped 15 seconds into the sound play. So we did this it helped us determine whether or not the fish were actually responding to the acoustic cue alone, or if they were just responding to the presence of bait. So it allowed us to kind of zone in on those important 15 seconds prior to the food drop. And during the next 10 days, we introduced the 2 different reinforcement methods that I mentioned previously, the continue and partial reinforcement treatments. So the continuous group continued to be rewarded during every trial, 15 seconds into sound play. And then the partial reinforcement group was only rewarded for 1 trial a day, chosen at random. So sometimes it would be during the morning trial, sometimes during the midday trial. So it varied every single day. And the reward was equivalent to all 3 feedings, which was 1% of the fish's body weight. So they were getting fed the same amount as the continuous treatment group, just all at one time. So next was the extinction period, where only the sound played and there was no food reward for either treatment group. Um, So the extinction period varied in length between the 2 rounds of the experiment. One lasted approximately 14 days and the other approximately 28 days. So we wanted to see how long it would take all of the tanks to stop responding to the acoustic cue. And then after all of the tanks stopped responding to the cue, there was a 7-day positive control period where the fish were fed 15 seconds into sound play again. And this was just to ensure that all of the fish were able to regain this conditioned behavior after the extinction period concluded. So every trial was recorded, and our team analyzed the fish behavior for a little over 2,000 videos between the 2 rounds of experiments. So when watching the videos, we examined the fish activity 15 seconds before the start of the acoustic cue, and then 15 seconds after the start of the acoustic cue, ending when the feeder dropped the food into the ring. So the training period was excluded from the analysis because the sound and food was dropping at the same time, and it was more to habituate them to everything. But then moving forward, we recorded the maximum number of fish displaying the conditioned behavior at one moment in time. So we define the conditioned behavior as the fish approaching the feeding ring, touching the ring, or signs that they were anticipating food near the ring. So this could be them orienting themselves with their snouts up towards the ring. So we chose this maximum number of the fish displaying the behavior at one time because we didn't have any tags or any way to seek out individual fish when they were out of the camera's field of view. So we determined that this was kind of the best conservative estimate of conditioning. And when we were analyzing the 15 seconds before the acoustic cue, we didn't really see the fish displaying any of these conditioning behaviors prior to the start of the cue. So it kind of confirmed that there was no temporal association with the feedings. So we have just recently finished analyzing all of the videos, so we haven't gotten into any of the nitty-gritty data analysis yet. So what we're seeing here are just some preliminary trends between the 2 treatment groups. So for this graph, each panel is a different experimental period in chronological order. So it goes from conditioning to reinforcement to extinction to positive control. And the x-axis is the number of days in that period, and then the y-axis is the average maximum number of fish displaying the conditioned behavior. So this is what our data table would look like. So the number of fish displaying the behavior was recorded for all 3 trials for all 20 tanks for approximately 2 months. For the simplicity of this presentation, we ended up averaging the maximum number of fish conditioned across the daily trials for each tank, which gives us this number here. And then we also averaged the average amount of fish displaying the behavior across the tanks for each treatment. So that's why we're only seeing 1 point for each treatment on the graph that I just showed you. When we dive into our statistical analysis, we won't be aggregating the data this tightly, but it's nice to see our preliminary trends. So for each panel, we could have a maximum of 3 fish responding or a minimum of 0 fish responding. And we see in the conditioning period, both treatments are following the same pattern. We're slightly increasing the average number of fish displaying the behavior as time goes on. And then when we get into our reinforcement period, we see this drop with the partial treatment group. And that makes sense because they're being exposed to a new feeding regimen and they're only being rewarded once out of the 3 daily trials. And then during the extinction period, it actually looks like the partial treatment group is continuing to respond longer and not losing the behavior as quickly as our continuous treatment group, which is interesting because that lines up with what we were expecting to see with the partial reinforcement extinction effect. And then when we go back into our positive control period, we see both treatments bounce back fairly quickly and begin increasing again at the same rate, more or less. So, like I mentioned, these results are very preliminary, so you can only make general conclusions from what we're seeing visually, but it seems like all of the tanks have developed the conditioning behavior in a timely manner, typically within 3 days, and that the partial reinforcement group is retaining the conditioned period or the conditioned behavior for the same period of time, if not longer than the continuous reinforcement group. So in the field, that could mean, cutting down on baiting every single day and lowering the costs associated with bait and labor. And then the next steps of our analysis will include further analysis of the treatment effect on the maximum number of fish conditioned for each individual period using a binomial regression model. So we will be zooming in and kind of looking at more of the detailed data. So moving on to our field study, which began in June of 2023, and it should be wrapping up this weekend. The goals were to once again test if carp can be conditioned to associate an acoustic cue with a bait reward in a natural lake setting, but this time with a higher carp density. And we also wanted to determine if the use of the acoustic cue in pair with bait could synchronize feeding aggregations, which could increase catch rates of physical removal strategies down the line. So our study site this year was Harrison's Bay, a northwestern bay on Lake Minnetonka, which is approximately 67 hectares. So it is a larger study lake than what we were working with previously. We again established 2 baited sites, which will be referred to as Site 1 and Site 2. And those sites were approximately 1,200 meters apart. And then prior to the start of the study, we electrofished along the 2 shorelines corresponding to our sites. Carp typically have narrow home ranges, so they usually don't travel very far from their kind of home shoreline. So that's why we just stuck to those 2 shorelines instead of electrofishing the whole entire bay. So we did 7 20-minute transects along each of the shorelines, and we ended up tagging about 70 fish per site. And we used PIT tags, so passive integrated transponder tags. So those are similar to what vets use in cats and dogs. And the average length of the carp that we were tagging was 660 millimeters, so approximately 26 inches long, the largest being 780 millimeters. And then we also sound mapped each site similar to what we were doing in our lab with a sound trap hydrophone to see how far our acoustic cue would travel. So the sound was measured in 45-degree increments from the speaker, and every 10 meters out until the sound was indistinguishable from the background noise. So the sound at site one traveled up to about 150 meters, and at site two, up to 100 meters. And these are both measurements from 90 degrees out, so straight out from our speaker. And there was no overlap in the sound field, so you can't hear the acoustic cue from either site, making them independent from one another. So here's some images of our two study sites. Both sites had a harder sandy bottom, an ample amount of lily pad mats, but very little other aquatic vegetation was present. And we had a UW30 underwater speaker that was suspended halfway into the water column and hooked up onto an amplifier on shore. So that's the same type of speaker that we were using in our lab setting. And you can see the little orange buoy over here. That's what we use to mark off our speaker. And then we also had a remotely controlled feeder, which was a 5-gallon bucket with a conveyor arm on the bottom that regulated the corn being released from a small hole. And then underneath our feeder was our pit detection antenna, and that was connected to our biomark pit reader and data logger on shore. So we were continuously scanning for tags, and we had a power supply of 12-volt marine batteries hooked up to our solar panels, so we had continuous power. And luckily, we didn't have any issues with our recording devices. So we have data for pretty much every single second for the past 4 months. We also, something new to this year was our remote system, which is this smaller solar panel and control box. So this system was actually hooked up to our amplifier, so it could control when sound was being played over our underwater speakers. And it was also connected to our automatic feeders through a cellular modem. So we had a website that allowed us to play the sound from a remote location or feed our fish, and we could select the amount of pounds that we wanted to feed them every day. So that was a really awesome addition to our field equipment this year. So the acoustic cue that we were using in our field experiments is very similar to what we were doing in our lab. So it was a 400 hertz pure tone, and it was pulsing for 2 seconds on, 2 seconds off, but we extended it from 30 seconds to 3 minutes just because we were in a larger space. And for all of the trials where we were playing the sound and dropping bait, we dropped 12 pounds of corn a minute and a half into sound play. So that was very similar to the lab study when we were dropping our food 15 seconds into sound play. That way we can kind of see the fish's behavior, in that first minute and a half. So here's an overview of our experimental timeline. We had a conditioning period that lasted 28 days. And during this time, site one acted as our control site and received only bait, which was cracked corn. And then site two was acting as our test site, and it received both the acoustic cue and bait. And then after a conditioning period, we moved into an extinction period, which was similar to the lab. We removed all food reward. So at site one, there was nothing occurring. And then at site two, we continued to administer the sound nightly. And then we had a 3-day break where we just did nothing at either site. And we kind of let the fish reset. And then we actually swapped treatments at both sites, and the whole entire protocol was repeated. So site one would then have our test treatment of bait and sound, and then site two would have our control treatment of bait only. So we're currently in that break for the second round of the protocol currently, and we'll be starting our positive control period tomorrow. So during this 3-day period, we're just going to reintroduce bait at both sites to ensure that attendance and consumption is still normal. So here we can kind of zoom into what our nightly trials looked like during both the conditioning period and the extinction period. So we had 3 trials each night, 45 minutes apart, and we alternated the start time similar to the lab study, just to prevent any kind of temporal association with feeding. So during our first trial for the conditioning period, we only played the sound at our test site, and we did nothing at our control site. So we wanted the first interval to serve almost as a dinner bell, to see if fish were to swim to the site on the onset of the cue before any bait was introduced. So it could help demonstrate potential acoustic conditioning. And then during the next two trials, we would play the sound and bait 12 pounds halfway through the cue. And then at site one, we would just bait 12 pounds. And during the extinction phase, like I mentioned previously, we removed any of the food rewards. So we were just playing the sound at site two every 45 minutes. So here we're looking at the entirety of phase one. So in this graph, we have the unique tags detected per hour on the y-axis, and then we have the dates on our x-axis. And we see at site one, we have average peaks, so the highest amount of fish, per hour are usually around 10 fish per hour, whereas site two, they are slightly higher, more so around 15 fish per hour. And at both sites, once we removed the food and we were just playing the sound or doing nothing, sorry, this shouldn't be here. They didn't experience any sound at this site. But we see a very similar pattern of extinction of the behavior. So they stop attending the site very quickly once the food reward was removed. We also see the feeding aggregations begin to tighten up as the conditioning period continues. It's kind of a difficult claim to make solely based off of what we're seeing visually, but this is an area that we will be doing further statistical analysis so we can see if one treatment kind of synchronizes and tightens up these feeding aggregations more so than the other. But just visually looking, it looks like when we have sound present with our bait, these daily feedings seem to be a little bit tighter than when we have bait alone. And then this image is the same as the graph we saw previously, but for phase two. So we swapped treatments at both sites. And again, I apologize. This shouldn't be there. This is a period of nothing for site two. So here we see that the attendance and the average peaks are similar to what they were during the previous phase. So they're similar. And we could potentially see the tightening of the aggregations maybe happening as well. It looked like these might be a little bit tighter with sound than without sound. However, again, we can't really make any of those claims quite yet until we do the statistical analyses to kind of back it up. So I know this graph may look a little busy, so bear with me. So here we're looking at the individual fish present during the nightly intervals. So these are all individual tag numbers. For the last 3 days of the conditioning period and the first 3 days of the extinction period. So these red lines denote one nightly interval. So remember that the first one is usually just sound alone and the next two are sound and bait. So this is just showing that we have a lot of individual fish data collected throughout the duration of our study. So when we're going to be diving deeper into our data analysis, we'll be focusing on these individual fish behaviors between treatments at their respective sites. So allowing or swapping the treatments across sites allows every fish to be its own control. So we're able to kind of expand our sample size, allowing us to examine the data under kind of a finer scope. And it's interesting because we can see typically in carp feeding aggregations, there's more dominant super feeders that are there pretty consistently. So we can see a couple examples of these fish that may dominate those feeding aggregations, whereas more fish or some other fish come more sporadically. And then once we hit our extinction period, we kind of lose attendance very quickly at our sites. So some primary conclusions that we can make based on our visuals is that carp did not respond to the cue alone in the study lake. And we saw that very clearly with how quickly we lost attendance during our extinction period. And also that sound is not deterring the fish away from our sites because attendance and average peak size is fairly consistent across treatments. So when we get further into our data analysis, we want to examine the distribution of the feeding aggregations to see if it's actually tighter and more synchronized when we use sound in combination with bait. And then moving forward, even sound may not be the clear-cut answer that we were hoping for when looking for solutions to enhance bait and removal strategies. But there is a lot of work that could be done looking at different baiting regimens like we saw in our laboratory study. So it'd be really interesting to take those ideas of continuous and partial reinforcement and bring them out into a field setting because it could have management implications down the line. And with that, I'd just like to thank MASARC and the Minnesota Environmental and Natural Resource Trust Fund for funding this project, as well as my project committee, seasonal technicians, and volunteers from Harrison's Bay. This project has had a lot of moving parts, and none of it would be possible without all of your support.